Hello, everyone. My name is Wisam Bazbaz. I'm a PhD student at Professor Haytham Amal Lab in the School of Pharmacy at the Hebrew University of Jerusalem. And today I will be talking about the role of thioredoxin in Shank3 mouse model of autism. Actually, thanks for Professor Barak for giving the introduction instead of me about the Shank3 mouse model. However, we use a slightly different mouse model. Okay, so I will briefly talk about autism. I think there is no need. Autism, as you know, is a neurodevelopmental disorder that affects 1 in 36 children in the US, and its signs start appearing at the ages of 2 to 3 years old. These signs include reduced sociability, repetitive behaviors, impaired verbal communication, intellectual disabilities, and sensory anomalies. Despite the severity of these symptoms, there is no biomarker for the diagnosis of ASD, neither there is an approved treatment for the core symptoms. Usually, physicians give some antipsychotics, for example, risperidone for the treatment of autism, but they have less efficacy and maybe 1,700 side effects. From here comes the urgent need for new therapeutic targets. So the cause of ASD is still not known. There are many factors that are thought to be responsible for this pathology, including environmental conditions like contamination, maybe the use of drugs in the pregnancy, and genetics. There are hundreds of genes are thought to be related to ASD. In our lab, we focus on 2 of the high-risk genes for ASD, which are Shank3 and SNTNAP2. This work was done in Shank3 mouse model of autism. As a brief reminder, Shank3 is a postsynaptic scaffolding protein in the glutamatergic synapses, and the knockout of Shank3 in mice induces ASD-like behaviors. So previously, our group intensively studied the role of nitric oxide in autism. We previously showed that in the Shank3 and SNTNAP2 mouse models, nitric oxide is highly produced. We quantified this by 3-nitrotyrosine, a nitrosative stress marker, and also by measurement of a direct metabolite of nitric oxide, which is nitrite. We showed that in the brains of these mice, the glutamatergic and GABAergic synapses are disturbed. You can see that the levels of NR1 and GAD1 are decreased in these mice. These are brain sections from the mutant mice, indicating that there are synaptic deficits in these mice, and also that the spine density is reduced in these mice. Spine density is a it directly describes the synaptic plasticity. All these changes in both mouse models were reversed by inhibiting nitric oxide synthase by a molecule called 7-9. Both of them are well-established mouse models of autism that exhibit several ASD-like behaviors or phenotypes, including reduced sociability, reduced social novelty, reduced novelty seeking, and elevated anxiety. So we wanted to check that this phenotype is mediated by nitric oxide. So we brought wild-type mice and we treated them with nitric oxide donors to see what will be the effect on the behaviors. And we saw that nitric oxide donors induced the same behaviors or the same deficits that we saw in the ASD mouse models, which include the sociability, social novelty, anxiety, and more. After we took the brains of these mice, we saw that there is also the same synaptic deficits that we previously showed in autism mouse models. So to conclude the previous findings, we showed that nitric oxide is a key factor or a key pathological factor in autism that leads to that is elevated in mouse models, in ASD mouse models, and leads to synaptic deficits and phenotypes. And we were able to reverse these phenotypes by reducing the levels of nitric oxide by a molecule called 7-9 that inhibits nitric oxide synthase. However, we don't know how this happens. The mechanism behind this is still not known. Not known. That's why we wanted to have a linker, which is a key regulator of nitric oxide, thioredoxin. Thioredoxin is a small antioxidant that works as a system, thioredoxin with thioredoxin reductase, its reducing enzyme, and NADPH. It suddenly stopped working. Oh. Okay, it is found in all living cells. It's highly expressed in the brain. That's why it is good antioxidant compared to other antioxidants that have less expression in the brain. It's highly expressed in the neuron, in the axons and in the dendrites, and it's involved in several neurological or neurodegenerative diseases and cancer. Behind its function as an antioxidant, it also regulates transcription factor activities, DNA synthesis, cell growth stimulation, and apoptosis. It does this by desulfide breaking and denitrosylation of S-nitrosylated proteins. By S-nitrosylation, I mean the nitric oxide post-translational modification, nitric oxide mediated post-translational modification. So many papers showed that thioredoxin can be a potential novel diagnostic marker for autism and also a therapeutic target. So based on what I showed you, we hypothesized that the excessive nitric oxide production that we found in the SHANK3 mice can lead to S-nitrosylation of TRX1, leading to its inactivation and decreased expression, leading ultimately to elevated oxidation and S-nitrosylation, which leads to the phenotypes that we previously showed. So to study this hypothesis, we measured the levels of the TRX system components in the SHANK3 mice, in the cortex of the SHANK3 mice, and we showed that the TRX system components are decreased, including TRX1, the cytoplasmic isoform, TRX2, the mitochondrial isoform, and TRXR1, which is the reducing enzyme. In addition, we measured the levels of peroxyredoxins, PRDX1 and 2, which are direct substrates of TRX1, and also oxidative and nitrosative stress markers. We validated the changes we found in thioredoxin also by primary cortical neurons. Then we used SHSY5Y cells that have the SHANK3 gene knocked out in order to assess TRX activity, and we showed that it is reduced in the knockout cells. Afterwards, we wanted to check or to investigate the role of thioredoxin in autism. That's why we inhibited it in wild-type mice, and we suggested that this may induce ASD-like phenotypes. So to do this, we used a molecule called PX12, an irreversible inhibitor, and it didn't only inhibit the activity of TRX1, it also reduced its expression. One suggested mechanism for this can be NRF2 system. NRF2 is the master regulator of antioxidants in the brain. It's responsible for the expression of over 250 antioxidant genes. So we expect when inhibiting TRX1 to have less induction of NRF2, leading to generally less transcription of antioxidants, including TRX1 itself. Indeed, that's what we found when we inhibited TRX1. We had less expression of NRF2. To conclude this slide, TRX1 inhibition leads to compromised antioxidant system, generally having less expression of antioxidants. Then we wanted to quantify oxidative and nitrosative stress with the treatment. We show the elevated levels of 3-nitrotyrosine, a nitrosative stress marker, and elevated PRDX1 and 2, which are oxidative stress markers that indicate that when inhibiting TRX1, we induce oxidative and nitrosative stress. Then we wanted to further investigate its importance in autism. So we mutated the active site, the cysteine in the active site, into serine, leading to its inactivation, and potentially leading to the loss of the reducing and denitrosylating function, leading to higher oxidation and S-nitrosylation, and higher oxidative and nitrosative stress. To validate that this mutation in the active site really leads to the loss of the TRX function, we measured the ratio between the reduced to oxidized ratio of PRDX1, a direct substrate of TRX1, and we showed that it is significantly decreased, indicating that the mutation leads to the loss of the function. Then we wanted to show that this mutation, how does it affect nitrosative stress, by quantifying 3-nitrotyrosine by Western blot and fluorescence, showing a significant increase. Afterwards, we wanted to directly quantify the nitric oxide. However, nitric oxide has a very short half-life. That's why we can't quantify it. So we quantified two of its metabolites, nitrite and nitrate, in the mutated cells, and we showed that they are elevated, indicating that the mutation leads to nitrosative stress. Then we wanted to show the effect of this redox imbalance caused by the TRX1 inhibition on the molecular and synaptic level. We quantified several GABAergic and glutamatergic proteins, including GAD1, the enzyme that converts glutamate into GABA in the GABAergic synapse, VGAT, which is the transporter for the vesicular transporter for GABA, GluN1, which is the subunit of the NMDA receptor, and two more scaffolding proteins. We showed that the wild-type mice treated with a TRX1 inhibitor have synaptic disturbance. The levels of these proteins were changed. You can see that GluN1, VGAT, and HOMER were decreased with the treatment. However, GAD 67 and PSD 95 were elevated. Then we wanted to assess the effect on the main phenotype of autism, which is the behavior. First, we conducted the three-chamber sociability test. In this test, the wild-type mice preferred to interact more with the mouse more than the empty cage. However, the knockout mouse of autism have no preference for the stranger mouse or the empty cage. And surprisingly, the wild-type mice treated with the PX12 have less preference to the social stimuli or to the mouse, indicating that TRX1 inhibition leads to sociability deficits. Afterwards, in the second part of this test, which is the social memory test, the wild-type mice prefer to interact more with a novel mouse or the new mouse that they never saw, more than the familiar mouse. However, the knockout mouse model of autism have no preference for the stranger or the familiar mouse. And that's what we also see in the wild-type mice treated with the PX12 or the TRX1 inhibitor, indicating that the TRX1 inhibition leads to altered social novelty. Lastly, in the object recognition test, where the wild-type mice prefer to interact more with the novel object more than the familiar object, the autistic mice or the autism-like mice or autism mouse model have no preference for either one of them, and that's what we also see in the wild-type treated mice, indicating that the treatment leads to reduced social novelty. So here we can see how much TRX1 is effective in autism, and inhibiting it can or may induce ASD-like phenotypes. So to conclude, in Shank3 mouse model of autism, which is a model with high levels of nitric oxide, TRX system is altered. When inhibiting TRX1 in wild-type mice, this can lead to redox imbalance, higher oxidative and nitrosative stress, and less antioxidants. This can also lead to synaptic disturbance and can induce ASD-like behaviors. I would like to thank you for inviting me, and my PI and all the group that contributed to this work, and of course the funders. Thanks for listening.